What is genome? Genome is the huge collection of genes. Genes are made up of deoxyribonucleic acid, which is called DNA. DNA is made up of paired strands that are composed of A's, T's, C's, and G's. The cells of our body are commanded by the code of 3 billion DNA. Cells make up tissues. Tissues make up organs. And organs make up organism, which is you. With DNA, we can analyze gene expression, which enables precision medicine, genetic testing, and species determination. So how do we get the DNA data? Sequencing. To sequence the first human genome, it took 20 years and over $3 billion. With the advance of technology, the next generation sequencing can sequence the whole human genome in a single run, and the cost can be reduced to $1,000. But there are some new challenges. The next generation sequencing first breaks the long string of DNA into tens of millions of smaller pieces. The small pieces of DNA are also called short reads. These short reads have to be assembled to rebuild the whole human genome by special algorithms, and then the possible mutations can be found. However, the NGS data analysis tools today are still not efficient enough to solve this problem. Even with high-end GPU, it still takes over 3 days to complete the whole human genome data analysis process. Therefore, we developed this System-on-Chip, SoC, which can complete the whole human genome analysis in 40 minutes with high precision. We also developed the customized graphic user interface so that the user can observe the analysis results directly. The DNA data analysis includes 3 steps: short-read mapping, haplotype calling, and variant calling. We will demonstrate these steps in this demo. Before we start, let me introduce the architecture of the SoC. In this SoC, we integrated an ARC processor, the proposed NGS data analysis accelerator, a DDR3 controller, PLLs, and other peripherals. The module on the top left side of the SoC is the DDR3 SDRAM. Besides, there are 3 daughter boards. From left to right are flash memory, JTAG interface, SD card, and UART interface. The personal computer transfers the pre-compiled instruction set into flash memory via JTAG interface. Before the DNA data analysis process begins, the test data are transferred from the SD card to the DDR3 SDRAM. After the data analysis process begins, all the analysis results and the processing statuses are transferred to the customized GUI by a UART interface. Due to the time limitation, we use chromosome 22 as test dataset. Here's our demo. The first step is the short read mapping. The information shown on the panel includes the progress bar, the length of short read, processed short read amount, and the total short read amount. After short read mapping is finished, the system proceeds to the second step, haplotype calling. In this step, the system assembles the mapped short reads by connecting the overlap parts. By this way, the possible DNA sequences of the subject can be rebuilt, and these sequences are called haplotypes. Besides the progress bar, the panel also shows the number of total active regions and the current active region. The system proceeds to the third step, variant calling. In this step, the differences between the subject DNA sequence and the reference sequence are identified and displayed on the screen. These differences are called variants. The panel shows the identified sequences from both the reference DNA sequence and the subject DNA sequence. The results of variant discovery are then stored in a standard VCF file. With these variant data, researchers and doctors can conduct further diagnosis and analysis. This is our demo. Thanks for watching.